Bel tengah record. 1, 2, 3. I think we'll start.
Okay. Assalamualaikum and very good morning to all. Thank you for joining us today in our first series of CODTIS Student Club. I hope everyone stays safe. So my name is Fatin and I will be assisting to moderate the program today. Alright, for today's Student Club, we have Ms. Ramya Devi, a doctoral student under supervision of Associate Professor Dr. Zain. Okay, so Ms. Ramya will share with us about her doctoral project and her recent findings. So without further ado, let's get started.
And the guys screen too. Nampak kan? Dekat screen tu. Okay, bagus. Silakan.
So good morning to everyone. First of all, please allow me to express my appreciation for the opportunity that you give me to join this journal club presentation today. And I hope I can make a good presentation of sharing my research project today. Now let me introduce myself to you just a little bit about myself. I'm a doctoral candidate currently pursuing on PhD, working on title effect of 1,4-benzalketone on bone marrow stem cells and hematopoietic progenitor cells via genetic and epigenetic mechanism under supervision of Prof. Madhya Dr. Zanandi Abdul Hamid. So we're moving to the next slide. The title that I'm going to discuss today is about my research project entitled Bone Marrow Oxidative Stress and Acquired Lineage-Specific Genotoxicity in Hematopoietic Stem and Progenitor Cells Exposed to 1,4-benzalketone. Before I discuss in further, let me summarize overall in terms of based on the abstracts. So I would like to emphasize few key points here from the abstract. First, the problem of the study. The main problem of the study is the hematopoietic progenitor cells are susceptible to benzene-induced genotoxicity with little information known about the mechanism underlying oxidative stress and DNA damage.
Salah. Sorry, minta maaf.
So then we go to the aim of the study to investigate the genotoxicity of benzene metabolite using the oxidative stress and DNA damage response on bone marrow cells and lineage-directed approaches. So the workflow involved using the control that is without the treatment, and the treatment with 1,4-benzalketone involve few doses starting from 1.25, 2.5, 5, 7, and 12. And then we proceed to the oxidative stress profile and DNA damage assessment on the bone marrow cells and progenitor cells.
Sorry ya, sebab awak tengok. Okay, macam mana saya nak? So dia macam saya nak bagi sekejap cutting.
Ini can make cutting as well.
That's why I nak try buat dia sebagai host cutting. Main screen.
You can try right click.
Kalau every time dia okay. See, itulah pasal masalahnya kalau duit.
So overall the results that shows for oxidative stress shows that there is an increase in the oxidative stress involving melaldehyde and also protein carbonyl, and decrease in the antioxidant activity involving GSH and SOD.
Sorry.
For comet assay assessment, it shows that there is an increase in DNA damage, which is more prominent in the myeloid and pre-B lymphoid, and followed by the erythroid cells. So overall, this shows that benzene able to cause lineage-specific genicity, targeting the hematopoietic stem progenitor cells. So let's go to the introduction. Just a brief introduction about what is benzene. So benzene is the most stable aromatic hydrocarbon, which is commonly found in industrial solvents, and it is a renowned common environmental contaminant. Humans are exposed to benzene through inhalation of automobile exhaust, cigarette smoke, and gasoline vapors. Yes, benzene is toxic. It has been classified as a Group 1 in International Agency for Research of Cancer, and chronic exposure of benzene has been linked to bone marrow depression, aplastic anemia, myelodysplastic syndrome, and leukemia. And it is well established that benzene exerts its toxicity through the reactive metabolite called phenol, catechol, hydroquinone, and benzoquinone. So a little bit more about the metabolism of benzene. So benzene is metabolized in the liver. It is metabolic activation occurs in the liver by few enzymes, and then it is carried to the bone marrow cells, and it is further oxidized into active quinone compounds by two enzymes, that is myeloperoxidase enzyme and NQO1 enzymes. So this metabolism of benzene is very important for bone marrow niche, and it is a target for benzene-induced hematotoxicity and leukogenicity. So what is hematopoietic stem cell and progenitor cells? As we know, our blood cells consist of stem cells which are replenished almost every minute in our blood. So actually it consists of stem cells which are able to self-renew to produce committed progenitor cells, and then which are able to differentiate to produce more matrix cells. It has been reported by previous studies that benzene metabolite causes damage in hematopoietic cells via multiple mechanisms involving chromosomal aberration, oxidative stress, DNA damage and apoptosis, alteration in the gene expression, error-prone DNA repair, and lastly, epigenetic regulations. So overall, this study is aimed to study the mechanism underlying benzene-induced hematotoxicity and leukogenicity targeting the hematopoietic stem cell niche, in which we involve different types of hematopoietic progenitors. So this is the experimental design in which we collect the bone marrow cells from the mice, and then we treat with a series of 1,4-benzoquinone doses. And then we proceed for oxidative stress assessment and also genotoxicity assessment involving the DNA damage. So as can we see, we isolate the long bone marrow from the mouse, and then we flush using the DMAM, and then expose to the bone marrow to 1,4-BQ for 24 hours. So this is the picture of isolation of bone marrow we have done in the laminar flow in order to flush the bone marrow cells to collect the hematopoietic stem cells. So based on the result that have been reported here is that there is a significant reduction in the GACs level and SOD activity upon the exposure to 1,4-BQ. And as we can see, it is started at the lowest concentration of 1.25. That shows that 1,4-BQ able to cause cytotoxicity even at non-cytotoxic concentrations. Next, when we look at the oxidative stress profile, which involving the malondihyde and protein carbonate, we're able to see that there is an increase in MDA and also protein carbonate. However, the increase is different according to between these two oxidative stress profile, in which malondihyde able to increase at 2.5 itself, but then protein carbonate at 5 micromolar. This shows that bone marrow cells are more susceptible to cellular membrane damage acquired by lipid peroxidation than cellular damage of protein via protein oxidation. And then exposure of high concentration of 1,4-BQ cause cytotoxicity, which reduces oxidative lesion as most cells undergo cell death modulated by the apoptotic pathway. So we can see there is a decrease in terms of the oxidative stress profile at the highest concentrations. So So overall, the oxidative stress can be explained by the BQ-HQ redox conversation in benzene toxicity, in which the HQ that is oxidized to BQ by myeloperoxidase enzyme actually releases electrons, and these electrons also called as reactive oxygen species, which actually increases the oxidative stress. And this indeed can cause increase in DNA damage in the cells. So let's look at the DNA damage profile on mouse bone marrow cells. As we can see, it increases in dose-dependent manner. So this increase in the DNA damage can be due to the increase in the reactive oxygen species, which is able to cause DNA strand breaks, DNA oxidation, and DNA adducts. So when we look at the different progenitors of cells, we can see that there is a difference in terms of the damage in between the progenitor cells, in which the myeloid and pre-lymphoid have to show more damage compared to the erythroid cells. So this might be due to the MPO and MQO1 ratio, which play an important role in benzene toxicity. So this is the picture of the comet X-ray. As we can see in the first picture, this is the comet picture, which we can see there is highlighted heat and also tail. And then the second picture shows that the comet is showing the DNA strand break. And at the third picture, we can see the extent of the damage, in which when there is the most damage, there is an increase in the comet formation. So this is the results according to the comet assay in between the bone marrow cells and also the progenitor cells. As we can see overall, we can notice that myeloid progenitors and pre-lymphoid progenitors show more damage compared to the mouse bone marrow cells and erythroid progenitors. So conclusion, 1,4-BQ exposure increased the oxidative stress in bone marrow cells via reduction of antioxidant capacity, GSH and SOD, and promotion of greater oxidative damage, NDA and protein carbonate. And 1,4-BQ exposure able to cause DNA damage towards the hematopoietic progenitor stem cells of different lineages involving myeloid, lymphoid, and erythroid. The implication of this study is that shows that hematopoietic progenitor cells may acquire different adaptive response or repair, which involve different survival mechanisms upon exposure to the drug. Therefore, further research on DNA repair mechanism and also the detoxification pathway, which involve both enzymes of NQO1 and MPO enzymes, is necessary to further characterize the mechanistic action among the different hematopoietic progenitors. So this is our team. So thank you.
Thank you very much, Ms. Ramya Devi, for the very comprehensive presentation. Let's open for any questions. So, you have any inquiries on the research?
Okay, microphone mute.
Okay, anyone have any questions?
The microphone is muted. So we need to open the.
Okay, so. You can also write in the chat if you have any questions to ask.
Cuba buka chat. Dia orang tulis tak ada kan?
masuk cepat. Tak ada orang tanya soalan. Janganlah, tak nangis sekarang. Sekejap. Semua ada bagi ke? Tak ada, semua ada. Sekejap. Okey, sekejap. Nak masuk. Okey. Kepada siapa yang dah hadir moderator tanya, ada soalan? Tak ada.
Kalau ada apa-apa soalan, kena semua.
Tadi ke? Tak ada orang nak tanya soalan ke? Tanya soalan dapat Rania bagi apa?
Bagi apa?
Tak pakai ke? Tak kan? Ada, betul tak ada soalan? Kalau ada soalan boleh unmute-kan mikrofon. Rania sekarang nak jawab soalan. Ada nak tanya soalan ke? Tak ada? Kalau tak ada, saya bagi QR code tu boleh scan. Tak boleh. Kakak tahu QR code semua boleh scan untuk termasuk Rania scan pun boleh scan juga. Boleh scan, sila scan. 1, 2, 3, 4, 5. Semua dah scan ke? Dimana dah scan dah?
Doktor, sekali lagi doktor.
Boleh tak? Siapa yang ada masalah dengan QR code, sila bagi tahu. Dah ke? Sudah saya nak tutup je skrin ni. Semua dah scan kan? Ada yang belum scan ke hadiran ke? Tuan Najib, thank you sebab datang. Tapi terlewat, tak apalah. Tuan Najib boleh scan Scan kehadiran. Kehadiran cakapkan ini kehadirannya. Untuk esok nanti saya datang. Nanti bagi tahu dekat kawan-kawan. Mana yang boleh kan. Itu saya. Okay. Jadi betul sesi dah habis kan? Okay. Sesi ni dah habis. Siapa yang dah scan kehadiran bolehlah tinggalkan bilik dan tinggalkan ruang virtual.